अब हम बात करेंगे कल्चर मीडिया की। द कल्चर मीडिया आर यूज्ड टू कल्चर द माइक्रो ऑर्गेनिजम्स इन द लैब। The purpose of the culture media is to provide basic nutrients to the microorganisms like bacteria or fungi. These basic nutrients include a source of carbon as an energy source, water, a nitrogen source, a source of phosphorus, sulfur, various minerals like iron, magnesium, or in case of fungi, some vitamins. Within the biotechnology lab, to culture the microorganisms, two types of media are commonly used. These are, broadly speaking, defined or synthetic media and complex media. Defined media, जैसा कि नाम से जाहिर होता है, इसकी कंपोजिशन का हमें पता होता है. We know how much grams of what nutrient is to be added. Like, for example, glucose is used as a source of carbon. Ammonium chloride or ammonium sulfate is used as source of nitrogen. And some other salts as source of phosphorus, sulfur or mineral. We have to supplement the media with certain vitamins if we have to establish a fungal culture. Complex media are often used in the routine lab. This is for the reason we prepare the complex media from the plant or animal extracts. चूँकि हम इन्हें प्लांट और एनिमल के एक्सट्रैक्ट से प्रिपेयर करते हैं, हमारे पास किसी किस्म की डेफिनेट कंपोजिशन इन मीडिया की नहीं होती, बट वी आर श्योर दैट all the basic nutrients will be provided by these plant or animal extracts. Complex media contains yeast extract and peptones in LB medium, which is used for the culture of E. coli, the bacteria. YEPD, as the name indicates, yeast extract, peptone and dextrose broth is commonly used for fungi. The dextrose is a source of carbon. Peptone acts as a source of nitrogen. फिर broth के हवाले से अगर बात की जाए, broth is simply a liquid culture medium in which all the nutrients are simply dissolved in distilled water. But sometimes we have to use the agar plates with the culture. For that purpose, the broth medium is supplemented with agar. Agar is basically a carbohydrate, a complex carbohydrate which is extracted from a seaweed, and it is used in the culture media because of its properties that it is solidified at a given temperature. It has no nutritive value in the culture media. Solid agar melts at 90 to 100 degrees centigrade. That is the reason whenever we have to add agar into the broth, we have to raise the temperature up to 90 to 100 degrees centigrade so that it can be dissolved. Whereas the liquid agar solidifies at 42 degrees centigrade. सारे process में when we add the agar, we plate the culture medium into the petri plates and then we leave them to cool down so that due to the properties of agar, the media becomes solid in the culture plates. Sterilization procedures or aseptic methods should be followed while preparing the agar media or LB media so as to restrict the culture of unwanted microorganisms.